Okay, well, hi everyone. Thank you so much for joining us today, both in person and virtually. We're very excited to present on our lovely internship here at ONRC. These are all of our ONRC interns, and they've created this presentation for you all. So with that, I'm just gonna let them get started and introduce themselves.
Yes, hello. So without further ado, after that bit of waiting, we are so happy to have you for this August 2024 Rosman Evening Talk. I'm Sophie.
I'm Olivia.
I'm Wyatt.
I'm Maggie. I'm Kate. And we're all interns from this field season, as Paisley just said. Today we'll be presenting you with some info about our summer of fieldwork here at the Olympic Natural Resources Center. But before we begin, we do have a couple quick notes for you. Firstly, there's a large collection of people here, both online and present, with a variety of different backgrounds. So we're going to be giving some background info on some of these forestry terms for context. So if anyone here is a pro forester, as a warning, we're going to be given some background there. Secondly, with each new location and work project, we use the art form of haiku to help us cope and reflect on our experiences. We've included these in the presentation. We hope you enjoy. And thirdly, we'd like it if you could save your questions until the end, just for the purposes of time. We'd be more than happy to answer those at the end, but just so that we have like a bit more of a grasp on kind of roughly how much time is taken up. And finally, throughout the summer, we worked on the traditional lands of the Quileute, Po, Queets, Yoncalla, Siletz, Umatilla, Hulapuya, and other tribes, a fact that was heavily informed a lot of these projects that we're about to list out to you. And one project that was particularly informed by this fact was, drumroll please.
Oh, I'm so sorry. I didn't understand.
I'm so sorry. Forestry field trials. Oh my goodness. So our haiku for this little project was high understory. What percentage do we pick? Learning plant ID. And yes, 575. For reasons you will learn in just a moment. But for, thank you.
I'm sorry, I'm just gonna get the hang of this at some point.
No, your question is. For some of the background on this project, the ethnoforestry field trials are a small scale study on a 5-acre chunk of DNR land out kind of northeast that way by the push. It uses people-focused forest management framework. So people-focused forest management framework, as you can kind of see here in this fancy little diagram, to manage forests for community needs, such as non-timber forest products like salal or understory species that can be foraged, as well as timber and wildlife purposes. So kind of focusing on all of those at once and using kind of fundamental kind of core values of learning, feedback, and all of that as displayed here. These needs were evaluated through a series of learning groups that our patron Courtney Bobson, who is on the computer right now, put on. And next slide, please. With this experiment was laid out kind of in this configuration. Understory species such as Nootka rose, Oregon grape, and whitebark raspberry, along with tree species, including Douglas fir and red alder seedlings, were planted in this configuration in these kind of squares. And so each of those squares that you can see on the map to the left represents one of the treatments here. And so these treatments, there's a couple different treatments that were utilized on these squares. There was fencing, there was spray like treatment to prevent wildlife browse. And then there was also different amounts of slash that were left on these plots. And so these different plots had these different treatments in order to study the effects of these treatments, which treatments produce less browse, which treatments produce the ideal conditions for the plants and the conditions that were highlighted in those learning groups that we talked about earlier. And the protocol that we use to collect data for this experiment is as follows. We collected the data to support this project that, again, our brave and lovely patron Courtney Lawson ran. The protocol for this experiment, number one, our mission was to locate the experimental unit. So we used that map that you saw on that previous page to navigate to the unit that we're taking data on. And you confirm it with each plot. There was like a little rebar that had a little tag. Just confirmed it, found the plot. Step one, check, complete. Step number two, we would take measurements on the planted seedlings. So I mentioned earlier, there were seedlings that were planted, Douglas fir seedlings and red alder seedlings. We would go through and we would take data, about to tell you what that was. But we take data on those seedlings that were planted previously that had tags to indicate that they were planted previously. And then step number three, we would also take data on the understory growth in certain understory plots. And so each one of those units, each one of those squares that was on that previous map has 6 understory plots. We would set them up, about to go through the protocol, but we would set them up and we would take data on those as well. So those were the 3 steps that we used to collect data. And to dive a little bit farther into the planted seedling measurements, step number 1 for that specific protocol for the planted seedlings was to find the seedlings. Oftentimes they were hidden by a lot of understory growth that kind of popped up since the planting. So at times it was a little bit tricky, but we would sort through, we would find the seedlings, we would mark to see if any had died. And so that was step 1, check mark on that. Step number 2 was collecting yes, no data, live, dead data. And another important part of this data was to see if the seedlings had been browsed. Like I mentioned before, a really big part of this study is fencing and light spray treatment. And so we would take browse data to see which of those treatments produced browse or less browse on those seedlings. So that was a big part of it as well. Step number 3, we would also take the diameter of the seedlings recolored using little calipers. We'd get down on the ground. One time we saw a snake crawl around one of the little bushes, or not bushes, the little seedlings. That was a highlight of that measurement. And then step number 4, we would also measure the seedling height using a little meter stick from Ace Hardware. Yeah, and so that was all the planted seedling data. Then we would move on to understory. As you can see in the 3rd step, we move on to the understory growth. And next slide, please. As you can see here, I'm so sorry. And with this one, step number 1 for that, we would estimate cover for each species present. So with this, we set up this little square. That's what the understory plots look like. We would look over it. We would identify which species were present. And then we would roughly see, that looks like it's around 80%. And we would give a percentage for each species. Step number 2, we would count the seedlings or saplings within that understory plot. And classify them into one of 3 height classes. So the really tiny ones were height class 1, middling ones were height class 2, on and so forth. And then we'd also estimate the percent cover of each of the following: wood, rock, bare soil, bryophyte, bowl, stump, litter, and basal rosette. And so bryophyte, for those who don't know, it's just like moss, pretty much. And then basal rosette, if you've ever seen the dandelion, like right at the bottom, the like really matted leaves and things that you can't really pull up, that's basal rosette. And so out of 100, we would estimate the ground cover for each of those. And then step 4, we would estimate the biomass. And we'll dive into that right now. Whoa, okay. So we would use some little tools called the biomass cube and the biomass booklet to estimate the biomass of each of these understory plots. And so we got to help use the biomass cube with Courtney and Stacy, who 2 of the people, Courtney, as we said before, she's on the Zoom. Thank you, Courtney, running ethnoforestry. And so the cube, essentially, you set it up It's a 3 meter by 3 meter cube. And you set it up, you take all of the biomass out, you like pull up all of like pulling weeds pretty much. And then you set that out to dry, and then you take the mass of all of that biomass. And that specific, having a picture of that cube area, as you can kind of see with this G1 photo, and having the biomass of that area allows you to estimate the biomass of similar areas, similar environments really easily without you having to pull up all of the biomass yourself for every single understory plot you take. And so that was super handy dandy. And you can see here a plot that would be quite similar to a page in the biomass booklet that would be able to very easily be estimated using that biomass sort of step. Yeah,
We created 6 pages.
we created 6 pages.
this summer.
Yes, we created 6 pages this summer using that. And we just would use previous pages and the ones that we created to perform that step. And then this, a little bit more expanded photo of those 2 pictures, or a little bit more expanded on those 2 pictures of potentially understory plot and a biomass page that you can probably use. That's my biomass. Yep. And so that was the biomass, that last step. But zooming back to the step number 1, estimating percent cover for each species present. A really big part of that is knowing what the species are and identifying the plants. So with that, plant ID time, we're gonna teach you a little bit of plant ID that we learned. And again, you know, we know a lot of you probably know a lot of these already. So for you, it'll be a review. But for those of you who don't, pay attention. There may be a pop quiz. Okay, so plant number 1, black-cap raspberry. This one's super important because this is one of the species that was planted in the ethnoforestry field trials. It is in the rubus family, and so this is a family that has salmonberry, thimbleberry, raspberry. And with these, you can kind of tell by that middle picture, there's 3 leaves kind of all together with these ones. Each ones are ovalish, a little spiky. They have the serrated edges and they're bright green on top, but then on the bottom, they're a kind of white fuzzy-ish. And they have woody white-green stems that you can see in that bottom left photo. Lots of spikes, not super comfy to walk through, as we learned doing field work. But the spikes, if you happen to walk through the spikes to get the berries, it pays off. These are some of the most delicious ones you can find. Quite delish, very tasty. They're reddish, purple, black, rounded, very round compared to a lot of other berries in the roots family. And you can tell them apart by the whitish bark. So that's the first one. Another plant, salmonberry. This is also in the roots family, Rubus spectalis. The leaves also kind of come in packets of 3, but as you can kind of tell from this one, if you took away that leaf, it's gone, pretend it's not there. It kind of looks like a butterfly a little bit. So that's a good, yeah, and then Rubus spectalis is its Latin name. It's spectacular because it's a butterfly. Yep. And so the stem is also, it's dark, it's woody, and then towards the bottom, it gets a little kind of peely, and it gets a little bit more spikes. But on the new growth, it doesn't have quite as much. So that's kind of a little bit about the stem. The fruit's pretty tasty as well, as you guys probably know, yeah. They're yellow or reddish, and they're pretty weakly attached to each other compared to some other berries. The little tiny parts of the berry, the little drupelets are. And fun fact, the sprouts can be eaten like asparagus. Yum. Okay, another plant, thimbleberry. We also have some samples for you guys to look at. We also have samples of salmonberry too. But here are some leaves if you want to feel them. These ones are light green. Yeah, thimbleberry, yeah. Pearl plant, oh my goodness. But they're typically about the size of a human hand, sometimes larger, sometimes smaller. Super soft and fuzzy. I've heard a lot of people refer to them as nature's toilet paper in the past because of this quality. They do not have spikes, they're unarmored, they're also, the berries are also very delicate. Oh, maybe those ones have spikes, those ones have spikes. The salmon berry, are you okay?
I was surprised is all.
I should have put a warning on there. Ah. Oh my goodness, yeah. But the berries for these simple berries, they're very delicate, rounded, bright red, taste kind of like pie, I would argue. And they also have very small, delicate white flowers, and fun little Latin lesson, part of florist means, small flower. And so it has a small flower. That's how you know. The next up, salal, also have samples of those, just gonna have some plants floating around. These samples do have some berries on them, so if anyone wants a little snacky, if, you know, do so, there's no risk associated with it, but you might not like the flavor personally. But the leaves for these ones, they're oval, they're evergreens, so you can see them in the winter too. They're dark green, finely toothed, and as you kind of see on the stem, the leaves alternate. The stem is also woody, it's also evergreen, and then the fruit. So you can kind of see there, it's blackish blue when it's ripe, 6 to 10 millimeters long, so small. And I think it tastes a little bit like the kind of artificial grape flavoring that you can get in like cough medicine and stuff. I love it personally. But yeah, tasty. And it grows either close to the ground, as a shrub or up to 2 meters tall. Pick from the plants. Fun fact. Another plant, sword fern. One of the reasons it's called sword fern is because the leaves are very sharp, like swords. It also looks like a sword, the fronds look like swords. But unitum, another Latin lesson. Unitum, I think, means something related to teeth. I think. I looked it up. But that's because the individual little leaves on there, they're really sharp, like teeth. And we have had some moments in the field, potentially falling into some very difficult to get out of, almost like they ate us. And the leaves, they're very long fronds, they can get up to 1.5 meters tall. They're also evergreen, kind of waxy as well, and they have large spore cases on the back, so you can kind of see the orangish. And the stem is usually kind of like orange and fuzzy-ish. They grow in clumps of up to 100. Whoa, big number. And the fiddleheads, the young fronds, they appear and unfurl in early spring. And story from the field. We found a wasp nest on one time. Bad news, not good. Yep. And then final plant that we're gonna be talking about, Nootka rose. One of the wild roses. Oh, I didn't fill out the leaf one. Ignore the top bullet point. The stem is woody, reddish, has spikes. The fruit, you need the rose hips. Good source of vitamin C if you ever want a little snacky. Not quite as tasty as the berries, but that's okay. And it grows in thickets, kind of on shores, forest edges, riparian zones, rocky slopes, a pretty wide range. Very pretty as well. Yeah, and so. Next page, please. Quiz time! If anyone wants to informally participate, does anyone remember what plant this is? Yes! Woo, round of applause.
And anyone on Zoom, if you know, please shout it out.
Yeah, shout it out, put it in the chat. What plant is this? Sword fern. Whoa! Yeah! And finally, this one's pretty tricky. Do you remember?
Salmon berry.
Ooh, close. Close.
Dingleberry.
Is that the black
Close.
cat?
Oh, exactly. Yeah, that one is tricky. A lot of the Rubus leaves look really similar. So final round, trickiest. You guys did it though. Round of applause for everyone for participating. Respect and admiration. Yep.
That's it.
Okay, amazing. So, after we kind of stayed here in, Forks and the extended kind of area, we went down to Longview, and that was kind of our next step along our journey. And the haiku associated with that has to kind of do with some of the work we were going, and doing down there. So, at last, KNLP, Longview, LiDAR, Alder 2, tree plots, here we come. So this was the, kind of the genesis of taking tree plots for us, which is exciting. Okay, so Longview, where is it? Kind of like to orient you guys in space up here, you can kind of see they're like the, where you are in the world. So we're kind of on the southern, kind of still western side of Washington, not on the coast, but near it. And there was 2 plots that we took data in. These were red alder plots. And there was one called Sprawl and one called Aldericious, which I think is a fantastic name. So Aldericious is this one that's kind of shaped like a top hat, I guess, or like a pilgrim hat. And Sprawl is this other little beautiful kind of diamond shaped one. And also here you can kind of see the polygons of them a little bit better. But these are the 2 plots that we were in Longview. Next slide. Sweet. Okay, so kind of the study that we kind of were taking data for, and the ideas going into taking this data and looking at these plots, they wanted to deal with 6 main things. I don't have my notes, let me grab those. So one of them was sustainable forestry and reduction of CO2, comparing red all versus Doug fir, and how they grow. Global warming mitigation potential, soil carbon accretion, so how much soil or how much carbon can accumulate in soil over time through the trees. And then mixed stands and crop rotations. And then another one was expansions opportunity, specifically because red alder is a deciduous tree. A lot of the forestry conferences, we do a lot of like evergreens and conifers. But these were some of the main 6 things that this report was looking at. The report title was called A Natural Climate Solution for the Pacific Northwest, ? And was looking at red alder. So we'll go off to the next slide. So this is what a red alder plot looks like. For me personally, this is one of my favorite parts along our journey, because before then we were in these areas that had been previously clear cut a long time ago. So there was no large trees, no canopy cover. And this was the hottest week, I think, of our entire journey. We were in the upper 90s. So it was kind of a blessing to just be covered in shade. But also the gorgeous green bright leaves that alders provide just like make the sunlight super bright. And it was beautiful. And that was some of us in action in the plots. So we'll go to the next slide. So a little bit about these plots. They were established in 1986 on Weyerhaeuser land. They were planted around 1986 to 1987, they think. So I think they're about like 37-year-old trees. But the land is now owned by DNR, and the trees are actually going to be harvested soon. So it's actual functioning plantation at the moment. So the plots are in Longview, as I said. And the objective of the Aldericious and Sprawl plots is to develop accurate height and DBH regression equations, which you might be like, what is that? And I'm going to tell you really soon. And we're doing that by correlating ground DBH measurements, which we all took, with the drone LiDAR height data. This info aims to evaluate the productivity, so how well the trees are doing, how they're functioning in their ecosystem, and see if they could be potential climate mitigation and used for helping the climate up. What's really critical about the work that we did was just improving these equations and models that are used in forestry. Because LiDAR is done remotely, we want to make sure that the data that the LiDAR takes is as accurate as possible. So we go out onto the ground and we call it ground truthing, meaning that the data that's taken from the sky, we actually go out and see that tree and make sure it's in the right location so that there's no like skew or variance in like how far off this data could be. But we'll get kind of more into the grits of it. Let's see. It's funded by the Nature Conservancy. And I think that's all I have for kind of like the broad picture of the plots. Awesome. Okay, so what is DBH? DBH stands for diameter breast height. So that means you're taking a measurement of a tree, so the diameter, how big it is around, at a standard breast height. So like for me, it's about here on my body, but for maybe a typical taller person, I'm a little vertically challenged. It might be more at their breast. This measurement helps us evaluate the productivity of the trees kind of over time. So let's say you wanted to take a measurement on a tree every year and you can see, oh wait, it's not really growing as fast this year. Why is that? Maybe there's a freeze, maybe there was a disease that came through. So these measurements can be really cool to look at in a temporal range. But who wants to learn how to take this measurement? Any volunteers? If you can add it to the resume, I got my resume. Okay, no, we don't want a full list, but how about instead, we'll show you, you want turn? Okay, come on up, come on up.
Oh, I thought I was gonna go out in the field and do it.
No, maybe we can schedule a time. Hang out. Okay, so let's say this is a tree, unfortunately, it's not high enough, okay, let's pretend we're shorter.
Somebody shorter?
Yeah,
My daughter is shorter.
your daughter.
One's shorter. This is big.
Yeah.
I'm shrinking and.
This is me and all my nieces that are 14 now. They're all like, we're taller than you, Auntie Oli. I'm like, okay, so what you're gonna do is you're gonna kind of hook the hook end into the stump. Let's see. You can kind of like get it in an area where there's already like a break in the bark.
I'm sure you can.
Yeah,
There's no from the other side too.
I think we got it. And then what you're gonna do is you're gonna wrap around the whole DBH tape all the way around. Now, what you want to do is make sure that you're perpendicular with the ground, so you're really level. So a saggy DBH tape looks like that.
To the ground, I think.
Yeah, horizontal, thank you. Oh my goodness, can you tell I'm nervous? And then you're gonna pull it tight. So pulling it tight was actually gonna get a more accurate measurement. You're gonna see that you can,
Yes, and then you put your glasses on.
then you put your glasses on and then you read right over here
Oh, over there.
what the
Why
measurement
not
is.
here?
Because over
Oh,
here is
oh, I'm
your,
sorry, I didn't see that.
Yeah.
Okay, yeah.
so that's kind of how you take a DBH. And then the tape is really cool. To get it back up, you just go like this.
Right.
All right, I'm gonna hand it off to you. All right, so I know my measurement in my mind.
Oh,
I'm gonna
okay, you
have-
want me to do it?
Now I'm gonna have you do it and see if we get a
I
similar.
wondered why I was standing here. Where it stays put. That's gonna hold.
Oh, she's a pro.
Now we have to go to right there, and then you have your glasses on.
Yes, I do.
Oh,
Yeah,
okay.
we got the same measurement. So this DBH, we would say is 30.
Even though we imagine DH.
Yeah. Yes, even though it's not technically a breast height. So we both got the same number. It's 30.9. And can we give a round of applause? You're hired. Okay, awesome. So that's one of the measurements we were taking out in, actually for the rest of the season. It was a very common thing we did. Okay, so I've talked about this LiDAR. You might be wondering, what is LiDAR? So LiDAR is a type of remote sensing. Remote sensing means that you're taking, kind of imagery, if you can think about it, from remote places. So commonly it's done through satellite, really way up high into the sky. It's done through planes, so a little bit lower, or drones is really common because drones are a lot more affordable. And so I think most of our imagery was done with drones. It's very commonly practiced. But pretty much a drone will come across and it has these lasers that it shoots down a laser, boop, and then when it touches something, it shoots it back up. So LiDAR stands for light detection and ranging, meaning that you're measuring the range that laser goes down and comes up. And you can tell a lot from this measurement. And you can kind of envision it. I don't know if any of you put your face in one of these when you were a kid or put your hand and make cool shapes. But this is kind of what LiDAR is like. You can envision every single pulse that goes down from the laser as one of those little individual points. And you get an impression of whatever you're taking the imagery of. So in our case, we're taking imagery of the forest, so trees. And if you look off to the photo to your right, you can kind of see what that would look like, in some software. And our brain is beautiful. We probably are looking at this and deciphering its trees, but really these are just points, little tiny points in space, in 3D space. We call this a point cloud. But from this, we can get really cool, information about, tree height and so forth. But here, let's go to the next slide and we'll show you the actual, imagery from our alder and sprawl plots. These beautiful images were analyzed and produced by Ali Kruger. Shout out to Ali, and they were actually flown by, make sure I got their names right. West Fork Environmental. So shout out to Chris Erickson for taking, flying the LiDAR for us. So this is kind of what the point cloud will look like. This is done with an elevation medium, meaning that the top is the red color. It shows that it's like higher from the ground, and as you go down the color scheme from yellow to green, the green is closer to the ground, which is a nice way to just like visualize it. But STEM maps are really cool. They can be used in forestry to make maps and models, to study for ecological purposes. Because a lot of the times we want to study something that's really far away, we don't have access to roads, or we can't hike out to this, but we can fly LiDAR and see what trees are there. You can see how big they are for maybe timber purposes, or if you want to conserve those areas without actually having to spend the money to send trucks out. And so taking these data measurements helps us get accurate models and maps and really hone in on the precision of them, because we go in there and do the measurements in person. But yeah, so we'll go to the next slide. This is what a STEM map looks like. Again, the red is showing high elevation, yellow is like kind of medium, and green is lower elevation. And each of these points indicates where like we think a crown of a tree is. And then the circumference around those points in the shaded circle shows kind of the canopy like range, I guess you could call it. But yeah, that's kind of a background of what LiDAR and DBH are, and this will kind of set the ground base for all the other plots that we are going to tell you about in the future. All right.
Okay. Our next project was called LTEP. And LTEP stands for Long-Term Ecosystem Productivity. And this is when we drove down to the Willamette Valley in Oregon, from Longview. And the main like driving question of this study is what factors influenced directly or indirectly by management most affect the long-term productivity of the land. And this was a bigger scale project than say the Alder or the Ethno Forestry projects. There's 4 sites all around the Northwest. One is in Saco, so close to here, and then the Sayusla on the Oregon coast, the Willamette, which is where we were, and then the Siskiyou, which is in more Southwest Oregon. So a big like range of different forests. Also a large temporal scale, so there's it began in 1995, and there's studies intended up to 200 years. So kind of the idea of LTEP is that forests take a long time to grow, trees get old, obviously, so it's often hard to study our effects on the forest in short-term feasible scales. So like, yeah, the kind of the point of this is to try to get more applicable results by looking at forest on these wider geographic and time scales, if that makes sense. Another cool component of this study is that it emphasizes cross-discipline collaboration between researchers, land managers. As you can see, there's a lot of different agencies that are involved. So yeah, it's pretty cool to get involved in. And we want to go to the next slide. The 4 treatments in LTEP were the controlled treatment, which is no human manipulation, just kind of let the forest grow. Mid-Seral, which is kind of like the classic forestry, Douglas fir plantation. Late Seral, which is kind of like active thinning of trees to try to get the forest to old growth succession kind of quicker than natural, you could say. And then early sterile, which is like a recently logged area. And then there's two different blocking practice, which is just kind of like categorizing different units into how much woody debris they have, which is important because woody debris affects a lot of things in forests, such as water filtration, the soil health, how resilient the soil is in a drought. And it also creates habitat for small mammals, insects, and those are the base of the ecosystem, and the food chain. Yeah, and then this is kind of just shows, a disturbed land to old growth forest, and then for the next slides, I have kind of examples that we've seen slash just around. So early sterile forests, these two images look different because they both had different disturbances that made them into an early sterile forest. On the left is ethnoforestry, which Sophie talked about. And that was logged, so not a lot of standing trees, lots of light, lots of different understory, pretty biodiverse. A couple saplings, yeah, and then on the right, this was in the Willamette, a lot of our work sites looked like this, which was really cool because a lot of these trees were like fully burned. So similarly, lots of light, lots of understory, but it just looks kind of different, which is kind of cool to see two different examples of that. Yeah. Next slide is a mid-sterile forest. So this is kind of like mid-sized trees are dominating. There's more light competition because the canopy is kind of closing up. There's less understory. As you can see, the top one, there's always a lot of understory. So that becoming a more late mid-sterile, but also a different species. It's the alder fox. And then this was in a redwood fox, which, we'll talk about later. But yeah, as you can see, canopy closing, it looks very different than so. And then finally, late sterile forest. Here, there's more canopy gaps, more down wood, more tree diversity, and yeah, so those are different types of forests. Yeah, so just kind of an overview of LTEP is we're trying to better understand how understory regenerates, how things grow back, how different treatments affect the tree and understory biodiversity, how the forest woody debris, like the fallen wood, affects a forest, and also just how trees grow back. And we can use these things to like figure out how to log things more efficiently and sustainably, as well as kind of figuring out forests on a more ecosystem, ecology perspective to build more resilient forests for humans again. Yeah. And then, you want me to haiku? Okay, this is our Willamette haiku. It's a little dramatic, as you can see. And it will be explained later, so I will shoot it now. Camping at HJ, huge flames on the horizon, 10 minutes, let's go. Which describes our experience. You can see the subtle hints going on.
Okay, awesome. So as explained in the previous slide, we had the good fortune of camping in the H.J. Andrews Experimental Forest while we were down in the Willamette Valley. And basically, a quick summary of what the H.J. Andrews Experimental Forest is and what they do. It was established in 1948, as you can see from this awesome sign, by the U.S. Forest Service. And originally, when they established the site, they had meant it for purposes of increasing forest operation efficiency. So a lot of their projects were geared towards road engineering and just forest regeneration. And then since then, the forest has been kind of used for a variety of focuses, and they've worked within forestry and also kind of expanded their horizons outside. They've worked on a lot of watersheds, and they have a lot of partners who study the salamanders and their movement in the area. So it was a really cool place to get to just be around. And when we talk about the large temporal scales, a lot of experiments, H.J. Andrews fits into that narrative. And not only are they kind of near the LTEP site, but they also have their own projects going on that also enlist in like a larger, longer term study period. And so, yeah, while we were in Willamette at the LTEP site, our attempted goal was to measure the 21 experimental units within the LTEP site. And so this is kind of a general layout of how one of our units would look. And so in each unit, which is like this bigger gray outline, there are 5 tree plots that were monumented and set up a while ago and then measured in 2012. However, because of the immense workload of 21 plots, or 21 units, we decided to take minimum of 2 tree plots per unit and 4 understory plots per unit. And we were particularly interested in measuring the approximate half of the units that burned during the 2020 holiday farm fire. So there's a lot of fire activity in the Willamette region. And in 2020, there was a pretty monumentous fire that burned through, starting by the Mackenzie River, centered around there, and then through parts of our LTEP sites. And we were very interested in, while it is a sad reality, and while maybe not all of the researchers who had put together the forest management plan expected this frequent of a fire regime in their future sites, it was very cool to see the kind of skeleton of where the fire burned, and we were interested in seeing the post-burn response in the overstory and understory, because that helps us understand kind of the effects that future fire regimes might have. It helps us assess fire tolerance of certain plants, what are pioneer species that come up after like a disturbance that might improve or just alter the general soil health overall. And it also shows us how, when trees come down and a greater amount of coarse woody debris enters the site, what that might do for wildlife, what wildlife might be attracted to the area, what wildlife might leave as a result of the fire. And so it was just a very cool opportunity to see something like that in the field. And then we also, to our tree plots that we started down in Longview, added 2 new measurements to observe these, this kind of like anomaly, and that was our burn severity and crown scorch. And so burn severity is, or crown scorch is the estimate of the percentage of foliage volume that was scorched or consumed during the fire. And so for this measurement, we were required to look up to the crown and see if the entire crown had been consumed, or rather that there were certain branches or parts of the crown still living and growing on it. And then we'd assign a percentage from 0 to 100 to kind of quantify that, and then understand on the broad scale what damage had been done and what trees had persisted through the fire. And then for burn severity, we have this description series from 1 to 5, and you can read a little if you're interested about the definitions that we used for each code. However, it was more so just a detailed classification of the burn on the crown as well. And then while in Willamette, just to understand general protocol, we had tree plots and understory plots. The 2 tree plots were set up using a circular plot system. So we went out to the previously monumented grid posts, which, if we could find them, not always the case, since there was a fire, so things could have been burnt, or an understory could have come through and hidden them. It was kind of a scramble, but when we did find them, we would go out from that and set up our circular 13-meter plots. And then we used our Javad, which is our satellite device, very nifty. You plop it in a part of open canopy, and then it kind of shoots up to satellites and gets a georeference, like pinpoint of where we are in space, just so that we can have a good read on the ground, and create these STEM maps, and use our Haglop device, which is our rangefinder that we would use, back ourselves up to the trunk of our trees in the plots, and then shoot at a transponder, which was standing in the middle, just this like kind of totem, if you can imagine, with a little birdie on top, as we call it, and it does this cute little chirp, which really exciting to hear when it first turns on, or also annoying, but we'd shoot at that, and then we would understand where the trees were, how the distance and the angle at which they were in relation to the center of the plot where that transponder was set up. And so we gathered that data just to kind of ground truth our LiDAR data that Ollie was referring to, and it just gave us a good like map of where we were. And then for data collection on the tree plots, we gathered various measurements on the individual trees, which included its species, the LiDAR visibility, which we kind of used just seeing like a general like look up and see if enough sunlight was capturing the crown, and if it looked like it wasn't being overcrowded by other crowns. And then mortality, if it's alive or dead, DBH, which you so masterfully represented. And then just our sort of anomalies, which is like reasons or damages, anything, an anomaly that might occur on the tree that would affect its growth. And so some of these include the tree being on a stump, which happens to a lot of our western hemlocks. They just naturally regenerate on those stumps, or maybe bear damage would affect, if there's bear damage present on a tree, then it would focus its energy on that, on healing that portion of the tree, so it might not grow as tall. Just things to keep a note that might be interesting in our data and our understanding of tree height. And then for our understory plots, our setup was a little different, and so Kate took kind of a big charge on this one, and she went out there by her lonesome, kind of got lost in the plots, you couldn't see her much, it was sad, like there's a bunch of understory covering her, but she was killing it nonetheless, didn't have to worry about that. And so she would take two PVCs and then set up 3x3 quadrat plots using those PVCs, and then she would do a scan over and document each and every species she saw in the plot, no matter whether it was one tiny little seedling or if it was a million of them. And then when it came time that she felt confident that she knew what every species was and that she had differentiated them, whether they're like in the same family but look alike, she would take the vegetation cover estimates that Sophie was talking about, so just kind of getting a representative overlook of what's in the plot and how much percent it's taking up. And then she would do a seedling and sapling count, which was just a look over and seeing if there were any seedlings coming up. And then ground cover estimates, which was referring to our rock, wood, bryophytes, just anything that didn't classify as vegetation, but still needed to be represented in the plot. And then she would just take our padlock device, which was the rangefinder, and kind of get the location of that in relation to everything else in our plot and to the middle transponder. And so this was our expectations. However, we had a bit of a reality crush, and while we were in Willamette, let me paint the picture for you. So imagine a bubbly and delirious crew after their first day in the field, babbling on their way home, scheming their plans for dinner, just talking as if they have endless time and as if the world is their oyster. And so we're driving home, and we all of a sudden, as we get into receptions, see both an alert that there might be something called, I think it was like a red. So it was like a red flash warning, so, okay, whatever, lightning, it could possibly be in the air, it's not the most concerning thing, but maybe for those of us camping, which we all were, not the best. And that was a little concerning, but we were always like, oh, we can go back up to our nice home, H.J. Andrews. And then unfortunately, about 5 minutes later, we see some smoke, it's not seeming as fun anymore. We do find out from an email that we're in a level 2 evacuation, also not the most exciting fact to hear after you're planning your dinner. And it just kind of, we should have predicted, seeing as there were a lot of signs, we hadn't heard of fires in the area. We did a fire training where we shot off little fire extinguishers the day before. There is premonition. However, it was, we were blinded. And so we drive up, smoke seems to be coming right from near our campsite. There's a blockade, we have to ask to get through to get our stuff. And then our colleague, Ali Cooper, who is one of the most like grounding, calm people, you just kind of see her go into Vin Diesel mode, like she's going fast and furious up those hills. And then I'm scratching my head, this might be a little more serious, okay? And so unfortunately, there were fire helicopters moving by. You hear the sounds. It was a scary sight. We pull up to our tent site. We have 10 minutes to go. Calling back to the haiku, 10 minutes, let's go. And if that was a challenge, we would have been the victors because everyone was out in 10 minutes. And it might have been because some of us from Washington have not experienced a fire before, a little, hey, fire under our butt, we were scared. And so we got moving, however, on our way out, my good friend Ollie, who was explaining the night before, just walking me through, kind of boastful, ooh, look at my walk-in closet that I've made myself in my campsite, do. Walk-in closet's still up, guys, that tent was not broken down. So then I see everyone run out of our car like they're agents in the war, and we got out of there. We got out of there safe. And unfortunately, this ore fire that had been the one that encroached on the H.J. Andrews facility did continue to spread in our time there. It's now sitting at around 2,700 or so acres. So it has gotten worse, but is seemingly sustained from the last report, or not sustained, but like not increasing in area since the last time I checked. And so that fire kind of prohibited us from our sleeping space. And then another fire, the little cowhorn fire, popped up when we were, because we're persistent. We were not going to just let this fire move us out of our work zone. And so we went back into the field the next day, and day after, unfortunately, another fire had sprouted up. That's why in this photo, we're all masked up, ready to go. And we learned a lot in the DNR safety training. However, we didn't learn how to drive over about 10 broken mirrors, which is what we must have done because bad luck was just our trend. And as you can see here, we had a tree fall down on our way. This is Kate, not on the day of her bee sting. However, I think the face is representative of what she might have been feeling after she sat in a nest and got 7 stings. And then, yeah, we had just, we lost the key, or didn't lose the key. Hey, sorry, our Hauser changed the key on us one. Just treachery. But at the end, we were able to still gather. We had a goal of getting a good, overview of the burn plots and the unburned plots, so we were still able to go in there. And I'm not entirely sure the exact number, but I think we did about 4 to 5 unburned and then a similar amount for burn plots. And we also learned a lot of valuable lessons. We learned how to move a tree off the road and the physics of that and the safety involved. We learned how to crisis manage, I think you could say. And yeah, it was tough. But we persist.
Okay, next is TP watershed experiment, not as exciting as that maybe. We spent a few weeks here doing a few different projects. Our haiku for this is high/slopes of doom, Sally's sisters on the move, Hagloff, where are you? So this is a collaborative study, led by DNR and ONRC, and it spreads over 20,000 acres, so it's massive. T3 refers to the smallest fish-bearing stream. And back to our haiku with these /slopes of doom, every site was super steep. So we got really comfortable with falling very quickly here. And then, so overview of the T3 watershed experiment, landscape scale management experiment to assess ecological, economic, and social impacts of standard and novel management approaches in upland and riparian forest ecosystems. So basically, this was taking in a bunch of different priorities and interests of different stakeholders, and trying out a lot of different prescriptions throughout this massive area of land. To zoom out, this is the large area of the study. This pink that is kind of hard to see, these are all the watersheds. And they also, all of our sites were backing up to streams and watersheds. And we spent most of our time at the bottom. Sorry, not good. Okay.
The red road is 101 as well.
So during our time, we spent time in 2 different prescription areas, doing understory plots. The first is the polyculture prescription, which is a mix of western red cedar and red alder, planted in different ratios. The second is EBDP, which is ethnoforestry variable density plots. And in those plots, it was Douglas fir planted in different clumps in order to help the understory, or to look at the understory and early sterile growth. Why these prescriptions? So for the polyculture, this was focusing on timber as well as different cultural uses of these species. It was ecological benefits for soil productivity and forest structure, and to create the spatial heterogeneity. And then for EBDP, this was looking at the understory, which is good for foraging for deer and elk, and also look to extend the early sterile stage to improve resiliency to the streams. So these are like the two different focuses of these that were taken by different stakeholders and different interests that people had. So first we did understory. In the understory, we did 30 plots in the polyculture, 10 for treatment, and about 30 in EBDP, also 10 for treatment. This is very similar to what we're doing in Willamette and in ethnoforestry, so I won't go super into the protocol. But these were 3 by 3 meter quadrat plots, just like Willamette, so pretty big. You're focusing on IDing the understory species, estimating ground cover, using that biomass booklet that we helped to create, and then also using haikus interlog. For the tree plots, we worked only in control plots. So this was to provide data to compare different tree treatments. In this, what we did to was fun is we monumented plots. So we got to set rebar at plot center, we got to nail in tree tags, and then also using Javad and Hagel to georeference our plots so that people could go back and find those plots and do more research later. Also very similar things that we did in other tree plots earlier in the summer. So we did species, DBH, live dead, LiDAR visibility, different anomaly codes, and then also distance and azimuth that we got from the haikus.
Okay, awesome. So our haiku for our last kind of step in our internship journey was with the Pack Forest Redwood team, I guess. And so this one is, let's take a tree core. Pack migrates like the redwoods, damage to code 80. Awesome. And so Pack Forest is the experimental forest in for UW, and they are just outside of Eatonville in a little forested plot. And so somebody forgot to mention about H.E. Andrews, they're also the experimental forest for OSU. This is kind of our equivalent of it at the University of Washington. And they're very dedicated to kind of helping students get experience on the ground with forestry, because it's something that I feel like is a little bit harder. There's not as many like opportunities out there. And as a result, there are some capstone projects, which is kind of like our culmination in the school for forestry and environmental science at UW. And so they were helping this student Hannah with her capstone project that centered around the redwoods and how we could compare the yield and volume of typical timber species like the Douglas fir to redwoods to discern the value of assisted migration and whether that would be a valuable resource, because the redwoods in California have been reportedly not doing as well, and so people are theorizing whether it would be smart to bring them up this way. And so I thought that was a really cool and like kind of new wave experiment that we got to take place in. However, we got to use a lot of like older forestry tools like the reloscope, and the reloscope is used to kind of view trees through it, and then based on the basal area of the tree, you decide whether that tree is in or out of the plot, rather than a set plot radius like we were doing in Willamette or Longview. And so we used that tool, and then we got to use these tree cores, which you can tell by these very muscular and cute interns and our crew lead. We would take it at DBH, and then we would screw the tree core in, and it was a bit of a task to yank it back out. I took a tumble trying to take off one of the parts. But once you got it out, you would slip it into a bag, and then Hannah is going to go ahead and count the rings and kind of do like a age estimation based off of that, which is cool. And then, just to close out our experience here, it's been so lovely, and it would not have been anything like this if we did not have all these lovely people to thank and all these people who helped us along the way, whether it be just providing support or information. And we would like to thank our magnificent crew lead, Paisley Bloom, for being so resilient and just dealing with every hurdle thrown her way. Super awesome girl. I think everyone can learn something from her. Also, our director, Courtney Bobson, always a big help. Awesome. Ali Kruger, who helped with the LiDAR imaging and came out with us in Willamette and Longview. Derek Peddle. Awesome. Nar. Jordan Sally. Anthony Odell. Brett Morissette from OSU. Gregory Edel, who came out with the Path Force team, and many, many more. Thank you, guys.
Here are just some fun photos that we have, different things that we did on the peninsula. Some of them actually aren't on the peninsula, but we did some fun backpacking trips. We camped on Second Beach, took some water samples.
There's Anthony
went to-
helping us
oh, yeah.
analyze our water sample.
We went to a lavender festival, but yeah, we've had an amazing time. And then here are also some fun things that we found in the forest. So different mushrooms, maybe a moss wig, some slugs.
Some slugs doing something.
We also went to the World's Largest Egg, which is pretty small, so we were very excited about it. Yeah.
And a really cool orchid.
Oh, yeah,
Oh, sorry.
native orchid to Washington.
Does anybody have any questions for us?
I have a whole bunch of questions, but I'll start with one, which is, I heard that Rainier is selling all their West End property. What do you know about that?
I have heard talk of it. Yeah, I was actually talking to my uncle about it, because he works in the forestry industry, and he was just, we're talking about how it's a lot of land.
I can't hear you.
We're just talking about how it's a lot of land, and it will be very interesting to see what happens, so I don't know if you have anything to add.
I don't know much about Rainier selling a lot of their land out here on the west end, to be honest. A lot of our work is like more in collaboration with like DNR, and we work almost exclusively on DNR land, aside from when we were in Willamette, we were working on Forest Service land, so I'm not super familiar, unfortunately. Maybe,
It announced the sale of about 100,000 acres along the north coast, up along, straight along, west of Port Angeles. That's, they've actually announced they're selling that, but there is rumors that they are planning to sell all the rest of their west end.
And will the buyers be forestry or?
Whoever comes, pays the most, I'm sure
Yeah.
that'll be what it is.
Thank you.
Basic question about the interns. Are you all from one school? Where are you in your educational process?
Yeah, we're all from UW. We're all at different stages. Like I'm gonna go into my senior year.
Okay,
Maya and I are going to our junior year.
So you're all undergraduates.
Yes,
Yeah.
and then
And then we have some
I'm
graduate.
graduating officially on Friday. I walked this spring and, yeah, me and Paisley and, yeah.
Oh, yeah, I walked, but I'm taking an extra quarter in the fall.
Yeah.
Yeah.
and I graduated in the spring.
Okay, so where are you heading then? Which? So you.
I don't know, this is the question, actually, I'm gonna take some time off and help Bam with some health things and then look for long-term job, hopefully in forestry, specifically in restoration work is what I would like to do. I'm really passionate about that, maybe in farming communities would be like an ideal landscape for me. Cause I love feeding people and I would love to feed people good food and restore the land as we do that. But yeah, I don't know, if you know of any jobs, let me know.
Ooh, yeah, I think, yeah, still figuring out the exact next steps, but I think eventually law school, like the litigation aspects of, I think a lot of this forestry have been really interesting. And I think like also making sure that the law is like accessible to folks, I think is another big thing. I think there have been like a lot of instances where the law has been very inaccessible. Yeah, so I think something in that realm, really. Very vague. Yeah. Paisley has big moves.
I don't know, big moves. I am currently also job searching, hoping to work in sort of the sustainable forestry area, looking to get more involved in both like management and research side and like dipping my toes a little bit into different areas of forestry and just getting more and as much experience as I can. Hopefully a job will come through. I just interviewed for one at the Snohomish Conservation District, looking for an AmeriCorps position. So I'm really hoping that I'll get that for the fall.
Any other questions?
Yeah.
I have a question for you all. Why did you come out here tonight? What are like the interests and like how did you hear about this type of like, this event, I guess? If anyone wants to share.
I'm here so I can make sure the data for Derek can work.
Oh, yeah, you love it already.
Always looking out for us.
Derek, make sure everything works around here. Big.
It's in the paper.
It was?
And it's a regular thing, used to be a regular thing.
We gotta get this paper, guys.
It's just a night out in Forks.
Yeah.
Yeah, we're out dancing, what we're doing here.
I'm always curious, I like to understand things, so. If you stop learning, you might as well be dead.
Yeah.
I think that's one thing we didn't have a slide for, but thank you all so much for, or we might. Coming out here, spending your time to learn about what we learned about. It's been so lovely to have you guys. Definitely feel free if people have more questions, but just really want to make sure that was said. Like, thank you so, so much for your time tonight. Thank you. Thank you.
Do I need the Zoom people?
Yeah, I was just to ask, Courtney, are there any questions in the Zoom?
There haven't been any that have come through so far, but folks are welcome to unmute and ask them or drop them in the chat. Now that I'm speaking, I'm curious, and any of you can answer, it doesn't have to be all of you necessarily, but I'm curious if there is something or more than one thing that you didn't expect to learn during your summer that you want to reflect on now.
Think about it for a second.
I did not think I would get so good at reading forest road maps and like learning how to navigate forest road and like call out on what we call a CD radio, which is pretty much you're communicating with logging trucks as they barrel down these crazy roads. So that was an interesting, cool new skill.
Which brings up a point, next year, there's gonna be a lot more training.
We had a training, I just didn't know we were gonna be training about it, but if that's good.
Here to drive the vehicles on the roads around here, 'cause,
Yeah.
some people have never been on this roads like this.
Totally,
It can be a little intimidating.
yeah, for sure.
It's kind of like say your next year.
I feel like it's like an aspect of it that people like don't expect at all too, just like basic clarification and things.
I feel like I spent a lot of time just looking at the ground at like small plants and understory plots, which is really cool. Just kind of get to know an area by the species that are there and kind of like the municipalities about that, which is really cool. And just getting better at planting is really fun because I don't see them in other areas. Just like get to know a place. Which is really
As a taxonomist, I appreciate that.
So the redwoods, I didn't know we had any out here until Greg Edel came over here and then I went out there to the site. These redwoods are this big around. Some of the cores, did you guys count any of the rings on them yet?
We did not,
I think they were planted around 30 years ago. I think,
yeah,
Greg said.
yeah,
Redwood goes really fast.
it does go very.
Yeah, but what I noticed, I saw about 10 of them.
Mhm.
And I think out of the 10, 6 of them had the tops blown. They're pretty soft.
I've got redwood on my land, my property, and the tops and do I go down a long time ago. No, it's just a hey,
They
mess.
kind of just, yeah,
Cathedral tops.
interesting to see them here though.
Yeah, that was very interesting.
Yeah, I wonder how well they're gonna do here because of that wind damages.
We have a
And,
couple questions on Zoom when you're ready.
Go ahead and tell us, Courtney.
Okay, okay, the first question is, oh, well, okay, I'll summarize. There's a couple of questions. The first question is, what does one of the attendees sitting in the room has a hat that says NIDYB? There's a question about what that stands for. And a second question about what animals, particularly bears or cats, did you all see? And I'm queuing you up for a great story that you didn't tell during your time in Willamette.
Would you like to?
I'm a ham radio operator and it's a vanity call. It's N1, it's just the area and the DYB are my initials, so, you know, just kind of mean.
Nice.
Who would like to tell our cool bear encounter story?
We just saw a bear in, Willamette, and it just scurried across the road from us.
Yeah, we were leaving the site from Willamette. This was also the same day I think that we encountered our fire.
Yes.
And I remember I was driving and this bear was going at full speed, like down the road, and like it was a pretty good slope, and it went like right by us, and it was a small bear as well, a little black bear. Yeah.
It was wild too, because not a second before we saw the
Oh,
bear,
yeah.
Maggie had just said, man, I like wish we could see a bear, like.
Yeah.
Yeah.
Sit on the road.
And said, as you wish.
We're like, Maggie, say more. What else do you want to say?
Other than that, we had no cougar encounters or other bear encounters, I think, out here on the peninsula. Lots of birds, lots of eagles, lots of slugs, lots and lots of deer for sure. We did a couple snakes, yeah, we did, yeah, a few salamanders. We saw some elk, but not really on our sites, but just like as mostly while driving, we saw lots of elk. Actually, in the redwood sites, we came very close to some deer, which was pretty cool. They kind of came up to us a little bit.
Lots of eagles.
Yeah, lots of eagles.
An owl?
Oh,
Oh,
yeah,
yeah.
that's not,
First day, I saw that mole.
yeah. That was sad.
That was really sad.
Any other questions in the chat, Courtney?
Nope, that's all.
Well, if there's no other questions, if we want to make space, if you guys have some, then feel free to come out and chat with all of us afterwards. We'd love to put names to faces if you guys have time, but we understand if you want to head out. Feel free to get refreshments, food and beverage, but thank you guys all for coming out.
Thanks, everyone.
Yeah, thank you for.
Thanks, Pam.
Thank you.
I'm just gonna.